Okay, so we're going to move on to aerobic cellular respiration, or the aerobic portion of cellular respiration. And it being aerobic implies that it uses O2 or oxygen, or must be in the presence of oxygen to progress. So the overall process of the aerobic part of cell respiration is pyruvate will eventually be converted into CO2 and H2O. So the CO2 is going to be generated in two separate steps, which we'll cover in detail. Pyruvate oxidation, which is kind of a preparatory step for preparing, you could say preparing pyruvate or preparing the remaining organic molecules for the citric acid cycle. Both of these are going to occur in the matrix of the mitochondria, or the mitochondrial matrix. So the energy stored in the covalent bonds between each carbon within pyruvate is actually going to be that energy, that's going to be the potential energy that's used to make the ATP molecules. And remember, those carbon-carbon bonds are going to be formed by electrons, which we harvest. Okay, so the first step is going to be pyruvate oxidation. Again, this is going to happen in the matrix of the mitochondria. And what's going to happen is pyruvate will eventually become oxidized. It's going to lose electrons. So pyruvate is changed from a 3-carbon molecule to a 2-carbon acetyl group, and then bound to a coenzyme A molecule. So pyruvate is oxidized into acetate or an acetyl group, and then that acetyl group is added to coenzyme A to form acetyl-CoA. And in that process, what we're going to do is we're going to strip away some electrons and plug them into NAD+, and then NAD+, while reduced, picks up a hydrogen, becoming NADH. So we have a reduced NADH as a product. And then also, if you remember, we're going from a 3-carbon molecule to a 2-carbon molecule. Where does that 1 carbon go, or how does it leave? It leaves in the form of CO2. So pyruvate oxidation doesn't create ATP. All it does is it creates NADH, CO2, and acetyl coenzyme A, or acetyl-CoA. And this process is going to require the presence of oxygen. Oh, I should have mentioned earlier that in order to get pyruvate into the matrix of the mitochondria, it actually takes energy. So it moves through the matrix of the mitochondria, or into the matrix of the mitochondria, through active transport. So it actually takes energy to bring pyruvate into the matrix of the mitochondria. Okay, so moving on, the reactants, the things going into pyruvate oxidation are going to be pyruvate, NAD+, and coenzyme A. The products are going to be acetyl-CoA, carbon dioxide or CO2, and NADH. So here's your 3-carbon pyruvate. Undergoing pyruvate oxidation, you're going from this 3-carbon molecule to a 2-carbon molecule, and then you bind that to coenzyme A, forming acetyl-CoA. Now what also goes into it is NAD+, which then receives electrons and a hydrogen, forming NADH, and that carbon from your pyruvate molecule is lost in the form of CO2. So this is going to be happening per pyruvate molecule. And remember, with glycolysis, you're making 2 pyruvate molecules. So you could actually multiply this by 2 if you're looking for the overall breakdown, or the products gained from the overall breakdown of glucose. You're getting 2 pyruvate, therefore 2 CO2, therefore 2 NADH, and therefore 2 acetyl-CoA per glucose. Now moving on to the citric acid cycle. So that's going to begin when that acetyl-CoA, that 2-carbon acetyl-CoA, is combined with oxaloacetate, a 4-carbon molecule, to generate citrate. So the name, the citric acid cycle, was probably inspired by the formation of the first product, which is citrate, a 6-carbon molecule. So then, to summarize the citric acid cycle, that 6-carbon citrate undergoes a series of reactions that involve molecular rearrangements, the stripping of more CO2 or oxidizing of it, And then eventually you form the end product, which is oxaloacetate, which then has the potential to bind to an acetyl-CoA and then start that process over. So it's a cycle. So for each turn of the cycle, what you're going to do is you're going to produce NADH, 3 NADH, 1 ATP, 1 FADH2, which is another electron carrier, and 2 CO2 per starting acetyl-CoA. So here we have acetyl-CoA entering, being combined with oxaloacetate, a 4-carbon molecule, to form citrate, a 6-carbon molecule. Then citrate is converted into isocitrate, alpha-ketoglutarate, and so on. And in the process, what you're doing is you're stripping away electrons, plugging them into NAD+, then, or NAD+ with electrons, then picks up a hydrogen, forming NADH. You're stripping away carbons in the form of CO2. And then here you're, again, stripping away carbons in the form of CO2. And by the time you get to this step right here, you've fully oxidized all 6 carbons of glucose. Because if you're counting glycolysis, which breaks a 6-carbon molecule into 2 3-carbon molecules, you undergo pyruvate oxidation, you strip away another carbon molecule from each 3-carbon molecule, you strip away another carbon from that original molecule, and then you strip away another carbon from that molecule. And so you have 6 carbons going in, and then through the process of aerobic cellular respiration, you have 6 carbons coming out. So essentially what we've done, by the time we've reached this point here, is we've fully broken down glucose, or at least the carbons coming out are the amount of carbons that have come in. Now we still have a few more processes to go. We actually shuffle around some more atoms. We form some ATP, or first we form some GTP, which we use to make ATP. And then we also strip some more electrons from that molecule, forming FADH2. And then we create some more NADH. Your NADH is actually going to harness a lot of potential energy, which will eventually cash out in the electron transport chain. So the respiratory chain, or the electron transport chain, is going to use your NADH and FADH2 generated by pyruvate oxidation and the citric acid cycle. And what it's going to do is it's going to create a concentration gradient of protons. And the final result is we're going to convert that concentration gradient of protons into ATP in water. So the active transport of protons out of the matrix into the inner mitochondrial membrane creates a proton concentration gradient, which I'll show you in a figure following this. The protons then diffuse through the proton channels down the concentration gradient back into the matrix of the mitochondria. The enzyme that's going to conduct that process is ATP synthase. So ATP synthase is an enzyme that synthesizes ATP. And it lets those hydrogens back into the matrix through a process called chemiosmosis to drive forward the production of ATP. So you're going to get roughly 34 to 38 ATP, depending on how you calculate it. It's going to depend on the conditions of the cell, as well as what energy usage you're considering when calculating the overall process. Because remember, to transport pyruvate into the matrix of the mitochondria, it actually takes place through active transport. So the respiratory chain consists of 4 large protein complexes, so they're going to be complexes of many proteins, bound to the inner mitochondrial membrane, but you also have these 2 other little proteins called cytochrome C and ubiquinone, which are intermediary molecules. They're not necessarily proton pumps, they're just electron carriers, you could say. They're these intermediary proteins that the electron will move onto and then move away from. So all of these proteins are going to help in passing the electrons along that electron transport chain. And then at the very end, what's going to happen is your oxygen, so this is where oxygen actually is used, your oxygen is then reduced. It's the final electron acceptor. And then it combines with 2 hydrogen ions to form water, sometimes peroxides, when the electrons eventually reach the final protein. So this is probably a better representation of what happens. rather than just words. Here's a picture. So you have NADH, which drops off an electron at your first electron acceptor, and this is actually a protein complex. What happens is that NADH drops off its electron, and that electron has an affinity to move from protein to protein to protein to protein, and so on. Now, here you have NADH filled with potential energy. It drops off that potential energy, or it converts, when it drops off its electrons, what it's going to do is it's going to convert that potential energy into the energy of movement. So this electron moving from protein to protein to protein to protein, that's a form of kinetic energy. And what we're doing here is, if you look at this enzyme, or you could say proton pump, here, what you're doing is you're moving hydrogens from an area where there is a low amount of hydrogens to an area where there's already a lot of hydrogens. You actually have a very high concentration of hydrogens, or hydrogen ions, or protons, in this intermembrane space. What you're doing is you're moving this hydrogen against its concentration gradient into this space, against its concentration gradient. And you're moving it against its concentration gradient with the use of energy, a form of kinetic energy. That would make this active transport. So you're using active transport to pump hydrogens from the matrix of the mitochondria into the intermembrane space. All of these hydrogens build up. That's a form of potential energy, because all of these hydrogens really want to make it back into the matrix, but they can't, because you have this membrane in the way. Now what we have here is an enzyme called ATP synthase, and what it does, kind of like a dam, is you let a little bit of hydrogen trickle through. So now you're allowing this hydrogen storage over here to dissipate, to disperse, to move down its concentration gradient. And it's going to move down, when it moves down its concentration gradient, that's a form of kinetic energy. So that kinetic energy, actually what it does is it physically churns your enzyme to allow it to fuse together ADP and inorganic phosphate to create ATP. So you go from potential energy stored in the form of an electron carrier to kinetic energy through the movement of electrons into a potential energy stored in the form of a concentration gradient into a kinetic energy, which is the movement of, or the flow of hydrogens through ATP synthase into, so you're converting that into another type of potential energy, a type of potential energy that we could actually use in the cell to conduct work, ATP. So here's just kind of a chart showing the change in free energy relative to O2. What you can kind of pull from this is that you have you could say that this first electron acceptor here has a higher electronegativity than NADH. And this protein here has a higher electronegativity than this protein complex. And this protein complex here has a higher electronegativity than this protein complex, and so on and so on and so on, until you reach the highest electronegative component, which is going to be your O2.